So, for my favorite brain structure project, I decided to talk about queer aspects regarding the hypothalamus. I chose the hypothalamus because in my research, I realized that there was actually more regarding sexual orientation and gender identity than I thought, and so that's why I chose it. And on top of that, I would also like to use this platform to talk about queer inclusion in the scientific community. In order to validate queer people's identity, we need to include them in the conversations regarding their own identity, and that means pulling more queer people into STEM fields. That means, being proactive and engaging with, nonprofits that focus on queer rights and, determining what is the correct rhetoric to use surrounding this and, just, using general empathy. Because, again, we all know, an academic community is supposed to be supportive, an academic community is supposed to be inclusive, but when we're leaving when we're leaving queer people out of the conversation, it means that, we're basically letting their identities be validated by whoever. And so with that, I would like to start out by talking about a study done by Alan Sanders at North Shore University's Health Systems in Illinois, where he identified on chromosome 13, located between the genes SLITRK6 and SLITRK5. I can't pronounce it, but it is spelled SLITRK. So these genes do regulate neural outgrowth, survival, and synapse function. But that really isn't my point here. I wanted to talk about how these are especially prominent in the diencephalon, which, as we know, is one of the structures located within the hypothalamus. And so it was found that in this study that certain components or certain variants of these genes, when activated, directly correlated with homosexuality and heterosexuality. And so I wanted to tie this back into LeVay's research about how the size of the hypothalamus relates to sexual orientation, and maybe how this gene expression correlates with size. So I thought that this was a very interesting concept. I also wanted to talk about how LeVay understood that prior to birth, there was activity that was very common in the hypothalamus that was pertinent to sexual differentiation. And so what we're seeing here with the expression of these genes is that determining what variant, depending on what variant it is, usually coincides with whether the individual in question is going to be homosexual or heterosexual. And so that carries me on to my next conversation about gender identity in the hypothalamus. And two Spanish scientists found that in 2013 that they used MRI on transgender individuals before and after hormonal treatment, and the results found that their brains were more similar in aspects to the brains of their experienced gender. And while that study was, conductive to conversations regarding gender identity, it was not zeroing in a specific part of the brain. And so I found that with this Dutch study, two Dutch scientists performed functional MRIs to examine how prepubescent boys and girls and adolescent boys and girls with gender dysphoria responded to adrenostenin. It is an odorous steroid that is known to cause a different response in the hypothalamus of men and women. And so they found in this study that adolescent boys and girls with gender dysphoria responded much like their peers of their experienced gender. And so these sex and this is significant because sex differences in response to odors cannot be influenced by training and environment. So there are no cultural stipulations or environmental stipulations behind this. It is clearly just biological, which again, that does support the idea that a transgender individual is biologically, predestined to be the gender that they feel. And this leads me to my third point, and it is oxytocin's impact on social phase processing and oxytocin's impact on social phase processing and the comparison of that between homosexual men and heterosexual men. And as we know, oxytocin is, the hormone that is common in social processes and sexual arousal and trust. And so it was found by Matthias Thintal, and others that sexual orientation does modulate oxytocin's impact on social appeal. And so it was found that homosexual men, when they received a dose of oxytocin through the nose, that they found male faces more attractive. But after heterosexual men received the dosage, it was found that they actually did not see any differentiation, and there was no effect on their approach tendencies. And so these results kind of give way to saying that homosexual men have a greater sensitivity to oxytocin. And to get down to like the hard data, heterosexual men gave higher ratings to female faces, but homosexual men rated both male and female faces in comparable fashions. And what this, again, like correlates to is that differential oxytocin action on brain structures, cultivates a more prominent behavioral effect of the hormone in homosexual men. In layman's terms, basically, oxytocin is just much more powerful in gay men than it is in straight men. And for my conclusion, I would like to point out that, again, clearly sexual orientation is a modulator of oxytocin's role. So whether or not you are gay or straight depends on how you perceive faces of the same sex, not only the same sex, but also the opposite sex. And in order to like piece together why gay men might react this way, they say, like I had read that enhanced sensitivity to oxytocin, might be an evolutionary strategy for survival because it reinforces same-sex partnerships. And thinking back in a time bound by agriculture and by roaming, these like reciprocal exchanges would be much more beneficial for people at that time. I would like to thank you for giving me this platform to talk about my favorite brain structure and how it impacts me and how it correlates with my identity. Thank you for this opportunity.